So, welcome back to the channel of Curious Corners in Biology, your own channel about curiosity in biology and scientific inquiry into the subject. So, today we will be discussing something interesting about cell membrane and endoplasmic reticulum. So, often you might have read about cell membrane, that cell membrane maintains fluidity and permeability. These are the two most important biological properties of the cell membrane. Now, how does it do so? You might have read that cholesterol has an important role in stability of the membrane, maintaining fluidity and permeability of the membrane. But the question is, how does it do so? So, yes, cholesterol has a role in maintaining exactly called as viscosity of the membrane. Cholesterol at high temperature fits in between the fatty acid chains of the phospholipid bilayer of the cell membrane. So, what does it do by doing so? It will simply restrict the movement of fatty acids in the membrane. So, the membrane is said to have become more rigid, more viscous. Because if the membrane becomes very fluid, then what happens is it becomes porous and susceptible to break. Okay, and if it becomes very fluid, then it becomes very permeable as well. So undesirable substances, undesirable small molecules can also get inside the cell. So at high temperature, cholesterol fits in between the fatty acid chains of the phospholipid bilayer and obstructs the unwanted movement to restrict the fluidity of the membrane. Now, what happens at low temperature? So, low temperature, the cholesterol is again going to fit in between the fatty acid chain of the phospholipid bilayer. Now, here at low temperature, naturally fatty acid chains would get clustered together. So, they will be unable to move. It is like they have been frozen. So, when cholesterol packs in, they will not pack tightly furthermore. Some spaces will be created, so at least the membrane will not become too rigid. When the membrane becomes too rigid, it also becomes more fragile and susceptible to break. So, there is of course a dynamic, a delicate balance in regulating the fluidity of the membrane to the optimum level which cell requires, because fluidity is directly connected to permeability. The second question which comes to our mind is, how does endoplasmic reticulum detoxify substances in the cell? Because often in books it is printed that yes, endoplasmic reticulum has a role in detoxification, especially the smooth endoplasmic reticulum. So, the process of detoxification involves changing harmful substances or water insoluble substances to water soluble substances, because water soluble substances are not harmful and can be easily let out through the kidneys of our body. And of course, hepatocytes or liver cells would contain maximum amount of smooth endoplasmic reticulum. Say for example, someone administers drug like phenobarbital. Now this is a drug which is used to treat epileptic seizures, but this drug can accumulate at toxic levels. So what inbuilt mechanism does our body have to remove the drugs, such sort of drug which has accumulated? So what our body does is that in hepatocytes, it is going to increase the amount of smooth endoplasmic reticulum. So naturally the surface area occupied by smooth endoplasmic reticulum would also double as the smooth endoplasmic reticulum has been increased. Now there is a system in smooth endoplasmic reticulum which is called a cytochrome P450 system, which is involved in detoxification. What this system does is converts the lipid soluble drugs or any toxins into water soluble substances to be let out easily from the body through kidneys. So hope this made you understand that how endoplasmic reticulum is involved in detoxification. So many such questions will be discussed further on. That much for today, and if you are interested in such content, check out the link in the description. Check out my book Curious Corners in Biology, available at Amazon, which is a compilation of such curious questions in biology in different topics of biology. Thank you very much. See you soon.